Okay, so my group and I did our research over CRISPR and how that researchers have been applying that to corals. Okay. Okay. Okay, so first, I'm gonna talk about how CRISPR first got like discovered and everything. It was first identified in repeated DNA sequences in bacteria. And it was thought to be part of an immune system that prokaryotes use uh, against infection from viruses. So how it actually works is the Cas9 protein, along with a section of RNA, form a complex, and that makes it able for it to enter the cell. And as the virus enters the cell, the Cas9 protein will cut some of that DNA's and it will like embed it into the own bacteria's DNA so that it will be able to recognize it again if it attacks and it will be able to destroy it. And yeah.
Uh, so this has a pretty big effect on, or coral has pretty big effect, or has a pretty big effect on marine environments and the environment overall. And coral actually uses photosynthetic algae inside of its cells in order to gain energy from those cells. And the warmer the ocean becomes, the corals will become stressed and the algae will die. And the loss of algae turns the corals white, called bleaching. And yeah.
So a lot of researchers have looked into this because corals are such a big deal. So they looked into specifically the relationship between specific genes and heat stress. Um, so as they're looking at all these genes, they realize how complicated it is. So what they did is that they increased the stress, they tried to see what mRNAs were being created so that they could see what genes were being expressed and not expressed.
So one specific researcher that our group looked into was the work of Dr. Philip Cleaves. And him and his research team basically really focused on heat shock factor 1, which is a protein that kind of relates to an organism's regulation of heat stress. And so it's really important in order to maintain homeostasis, and it's found in tons of organisms, including humans, but it's also seen in close biological relatives to corals. So an improperly functioning HSF1 protein can cause cell death from heat exposure. And basically what HSF1 does is it facilitates the presence of heat shock protein, and that works to correct the protein misfolding through homotrimerization. And so both HSF1 and HSP are very important to kind of maintain a heat tolerance. And there are a lot of genes related to those, but it's uncertain like how many and which ones specifically work to increase the heat tolerance of corals.
Lastly, I'm going to talk about the applications of using genetic engineering in wildlife conservation. So it can be used in a broader context for other wildlife. We can be seeing that in this specific case with corals, it was important to identify the genes that might evolve in order to help corals adapt to climate change. And looking at the role HSF1 plays in heat stress regulation, it gives promising signs of helping conserve these corals. CRISPR can also be used for other organisms to similarly identify the role certain genes play and possibly modify these genes or correct these mutations. And by doing this, it gives certain species and populations that are more at risk the ability to thrive and allow for the conservation of certain species and populations. And these are our resources.